I'd like to introduce today's speaker. We have Dr. Sandipa Gautam. She's an Cooperative Extension Area Citrus IPM Advisor. She is with the UC IPM Statewide Program Operations and UC Agriculture and Natural Resources at the Lindcove Research and Extension Center. Today, she's going to be speaking on managing mealybugs in California citrus orchards. So now I'm going to go ahead and pass this over to Sandipa. You can go ahead and share your slides.
Thank you, Cheryl, for a nice introduction. To all of you who have joined, good afternoon. My title today says Mealybug Management in Citrus Orchards in California, as I'll be sharing results from studies conducted under California conditions. But the concepts that I'm going to share with you today are applicable to mealybug management in other areas as well, other states, other countries as well. So let's begin. Here is the outline that I'm going to follow today for the next 45 minutes. We will begin with mealybug in citrus and why they are becoming a concern, especially for our California citrus growers. Then we'll talk about mealybug species, biology and identification, seasonal phenology, scouting and monitoring, and also take our time to learn about why all of this information is important to build and a sustainable integrated pest management program for citrus mealybug. And finally, we'll spend quite a bit of time talking about management options and best management practices for citrus mealybug. Most of the research data that I'm going to share today is the research conducted in the last 2 to 3 years. First of all, what are mealybugs? Mealybugs are soft-scale insects. They are sap-sucking pests in the order Hemiptera, belonging to family Pseudococcidae. Their characteristic feature is that they have soft, oval, shaped body, and they are distinctly segmented, and they're covered in mealy wax. These all the mealybug species have filaments extending from body margins that are covered in wax. And these filaments can actually be used as an identifying character for identifying different species. They are common pest problems in greenhouses, and management can be difficult due to their clump distribution. When it comes to host strains, they're quite polyphagous. They affect all kinds of crops, including tubers, vegetables, fruit, and ornamentals. And when it comes to fruit, citrus is one of the preferred hosts. And in citrus, they can be a problem for these two major reasons. The mealybugs, as sap-sucking pests, can cause direct damage by feeding. They extract sap from the phloem, and that reduces plant vigor. They can, infest citrus so much that they can actually kill the plant. They feed on all parts of plant. They feed on leaves, twigs, branches, fruit, etc. On leaves, their feeding can cause discoloration, bleaching spots, and leaf drop up to 80% has been reported in some countries. On fruit, their feeding would cause bleached spots, as you can see on the photo for lemon here. And their feeding can also cause abnormal shapes in fruit, especially in mandarin and easy peel varieties. And if they're feeding on peduncle, like the photo that you see on the right side here, it can lead to fruit drop. Up to 100% fruit drop has been reported where mealybugs are not managed. And because they feed on phloem, they tend to produce a lot of honeydew, and sooty mold grows on this honeydew, and presence of honeydew and sooty mold at the time of harvest can affect marketability of fruit. In California, 4 different species are reported. I got this information from UC IPM guidelines and from a publication done by Barrett and Lloyd in 1958. There are 4 different species as listed here. Each of them have distinct characteristics. The scarlet mealybug has dark red body expression. Their hemolymph is dark in color. Comstock mealybug, the second one, has a thicker wax covering and relatively longer setae at the base. Long-tailed mealybug, as the name suggests, has long filaments. But the most common species that we find in California citrus systems is citrus mealybug, Planococcus citri. It has pinkish body and somewhat brown, line that runs through the center of its body. Citrus mealybug is not only common in California, it's most common mealybug species in greenhouses, and it's present throughout the world. In California, I got these reports from CDFA reports. They were reported in Riverside in 1900s, in Ventura County, 1900s. Similarly, they have been reported from multiple counties in California. Now, these are samples sent by growers for identification, but despite mealybug being present, they have never been a concerning pest, an economic pest for citrus grower in California until 2019-20. When we look back in literature, what was done, literature published about 100 years ago, this was 1920, a monthly bulletin by California State Department of Agriculture. It talks about a success story that followed release and establishment of mealybug predator, Cryptolaemus montageri in this case. That was one study 100 years ago. Another 50 years later, similar success story, but for a parasitoid, Anagyrus pseudococci. Both these articles were based on studies, work done in Southern California. But now most of the citrus growing acreages is in the San Joaquin Valley. Nearly 75% of the acreage is in the San Joaquin Valley. And citrus mealybug has become a key pest for our growers. As you can see, it is IPM's most wanted, as seen on the Citograph cover page over here. Okay, let's talk about why. Why is mealybug becoming an increasing issue? There may not be one clear answer for this question, but there are several plausible factors that may have contributed to rise of mealybugs as a pest of citrus for our California growers. The first one, timing of loss of chlorpyrifos insecticide in California parallels with the rise of concern of mealybug as a pest. Chlorpyrifos or Lorspan was used for controlling California red scale, and that to some extent may have helped keep the mealybug population in check. Chlorpyrifos was also one of the effective ant control material, and ants are known to attend mealybugs, farm mealybugs, defend mealybugs from natural enemies, and also help in their dispersal. So ants becoming more of a problem may have something to do with mealybug also rising as a pest of concern. Second could be reduced use of systemic insecticides such as imidacloprid. Third plausible reason is use of broad spectrum insecticides and its effect on natural enemies, especially early season application targeting katydids and citrus thrips. As you all know, citrus thrips are key pests in California because they cause scarring. And 2-3 years almost for the last decade, they have been increasingly challenging to manage, and our growers have gone back to using broad spectrum insecticides, and that might have disrupted natural enemy balance for mealybug, and over time lead to mealybug population explosion. Another reason, late season application for pests like fuller rose beetle. Also, we can't ignore the fact that mealybugs are very cryptic in nature. They hide. They're not easily detected unless there's thousands of them crawling all over. They're easy to miss. Early detections, it's really easy to miss, and the pest kind of develops in the orchard, and by the time you notice, there is already too many. Weather also has a role to play because, if we have good, cold winter temperatures, there's a lot of overwintering mortality, but we haven't had that in California for almost a decade or more. And warm winter means more insects survive and slowly their populations develop. So all these reasons combined might be why we are seeing citrus mealybug population increasing in California. And as I was saying, mealybug infestations are hard to control because of several reasons. If you look at the arrowheads on these photos over here, each of these red arrowheads is a citrus mealybug with a female producing eggs with an egg sac. And these are really easy to miss unless you are in the canopy looking for them. These are easy to miss. Also, that green fruit over there, if you look at the navel, there is white stuff in there. That's a mealybug, probably a female with an egg sac, another similar, white spot. It looks like spider egg or bird poop in there. You miss this, give it 2 months, it can look as bad as this because mealybugs are highly prolific, and there can be frequent overlapping generations. Eggs are protected within the egg sac. The egg sac is hydrophobic in nature, so all these insecticides that are water soluble um, are repelled by the egg sac. Eggs are somewhat protected. We don't control eggs, and then reinfestation happens really easily. So 2019 is when a lot of you started seeing mealybug, which has constantly increased as shown in the survey results here. And there was in 2022 a growing need for an integrated approach to mealybug management. And this was due to the fact of there was increasing reports all over the San Joaquin Valley in all citrus varieties, and some of you started treating in 2021-22. But because this insect was never a pest in California citrus system, it was one of the understudied pests. We didn't know a lot of things about citrus mealybug. So in 2022, we went to Citrus Research Board, and Citrus Research Board funded a project to study its biology, its seasonal phenology, and figure out near-term management strategies for citrus mealybug. We first started with some basic questions. How does citrus mealybug look like? How much do we really know about this bug, and what is needed for research? First, we needed to know about the biology, life cycle, its stages, and identification, what species are present. Second, seasonal phenology. When does the activity start? And this is most important from management perspective, because if we don't know when the pest is active, then we can't develop a good management strategy. How many generations are there? What is the generation time? How do we scout for it? When do we look for mealybugs, and where do we look for mealybug? And how many is too many? And, with all that information combined, what are the tools that are available that works on mealybugs, biological, cultural, chemical options, and how to use them effectively? So let's start with the biology. From what's published in literature, life cycle of citrus mealybug begins as an egg. Egg-laying females can lay up to 600 eggs in a lifetime, usually in batches of 5 to 30. Within the loosely held egg sac, as you can see in the photos over here, that cottony looking, stuff underneath the female mealybug on that first photo is eggs. They are amber color, as you can see on the other photo. And then when these eggs hatch, the growlers emerge and they disperse. They start feeding. After they start feeding, they start producing these mealy wax that gives them a dusted with a flower appearance. And the females go through molting and become second instar, continue to feed, go through molting, become a third instar, and stays third instar until they're mated. Males, on the other hand, go after they turn into second instar, they make this loose cocoon and go through a short prepupal phase and then pupate and emerge as adult and find a female and mate. It's mate with the third instar, and it's only after that third instar has been mated, it becomes an adult female, start producing eggs. And that whole life cycle can take up to 30-35 days at room temperature. How do we identify mealybugs? As I mentioned before, there are a couple other insects and also different species. So mealybugs, they are soft scales. Their body is soft. Their shape is oval and pretty much flat. And the most identifying character is they're clearly segmented, and there is these filaments that are coming out of each of the segment. Especially for this species, Planococcus citri, there is a vertical line that runs through the middle of the body. As you can see on the photo over here and on the photo down, with the male too. And like I was saying, at each segment, there is a pair of setae coming through. On other species, the length of the setae would help you identify what species it is. But for the citrus mealybug, the setae is not as long as some of the other species. The immatures and adults, they have similar appearance. They look the same. It's just that the adults and, second and third instars are larger compared to first instar. Males, because they go through complete metamorphosis, they have wings. The wings are there's a pair of wing and have minimal venation. So there is no veins, not a lot of veins on the wings, and they have hairy antennae. And one of the other identifying characters for citrus mealybug males is that they have this tail filaments clearly visible sticking out, from the end segment of their abdomen. So how does knowing about pest biology help manage them? Let's take an example of eggs. So early in the season, when the population is mostly overwintering females and eggs present within the egg sacs, does it feed? No, it doesn't. Does it move? No, it doesn't. How do we monitor for it? We look for egg sacs near females. We look on the trunk. We look between clusters. We look, on fruit, on the navel end. That's where they're most likely overwintering. The chemicals most likely will not work because the eggs are protected within the egg sac and it's hydrophobic. So chemical management may not be an option. But Cryptolemus beetle are known to prefer feeding on eggs and females producing eggs. So, if you have a population that consists of mostly eggs and you want to do something right away, biocontrol might be an option. If you want to spray, maybe wait out, depending on temperature, a couple of weeks or a week in summer to spray chemicals, because crowlers and second instar, they move. They have waxy covering, but it's relatively less thicker compared to adults, and they can be controlled with contact insecticides and systemic insecticides. And for biocontrol, there are predators and parasitoids that will feed on them. For adult females, they move. Feeding, it's not very well understood. Chemicals, they have thicker covering, so may have little effect, but the predators and parasitoids, especially Cryptolemus, feeds on them. For adult males, they fly, and they can be monitored using trap cards. Chemicals, they don't live for long, probably for 2-3 days, so there is no point. But mating disruption is a potential for, confusing males so that they don't find the female and mate with it. Okay, that's how we would use biology to inform management. Now let's talk about seasonal phenology. When we started hearing complaints from pest control advisors and growers about citrus mealybug, we had no clue how mealybugs were moving in the field. When were the generations emerging? How many generations were there? The only reference was vine mealybug, which has 2 generations in a year, vine mealybug infesting grapes. And it would go under the bark, under the ground. So all of those information was available for vine mealybug, but we knew very little about citrus mealybug. So we started monitoring 13 trees in a block that we knew had infestation in 2021 because we wanted to know how mealybugs were moving. We started with double sticky tapes. We wrapped the inner branches and trunk with double sticky tape. As you can see in the photo over here, at the arrowheads are double sticky tape. And then we also placed cards and pheromone lure to attract males in this block and change those weekly. And we also counted number of mealybugs on leaves and fruits throughout the season. After 1 year of monitoring, this is what we have, the monitoring data. So let me explain this graph a little bit. On this graph that you're looking, there are 2 different y-axes. Y-axis on the left is number of citrus mealybug on a sticky trap per tree, the ones that we caught on the trap every week. And the bar charts are what is showing you that data. On the right y-axis is the number of males that we caught in the traps, and the dotted lines are the data for number of males per trap. And on the x-axis is the dates of trap collection. It's a 2-week interval. So what we found was citrus mealybug activity started in March because we caught crawlers, darker bar, in last week of March. 2 weeks later, we saw that males had started flying the first male peak, and soon after we started catching adults. So about 2 weeks after catching crawlers, we were catching adults on the tapes. And then that continued for almost a month. And then in June, we caught another population. And then there was big crawler emergence. And then we caught very little mealybugs on the sticky tapes. For the males on the trap cards, we saw the first flight in first or second week of April, second on last week of June, and then every month. So we saw 6 different peaks, first in second week of April, then mid-June, then mid-July, then mid-August, then mid-September, mid-October. So 6 different peaks. Now, the male data tend to suggest that, oh, the numbers are increasing, the population is increasing because we're catching males, but we don't find crawlers or females on our sticky traps. What happened? What happened is mealybugs moved to the fruit. And this data over here shows you percentage fruit infested with mealybug over months. And as you can see, in July, there was exponential increase of fruit infested with mealybug that kept increasing. So one of the things we learned from this data is mealybug, once they find fruit, they don't move. They sit there, they stay there, they multiply there, they have overlapping generation, they kind of, stay on top of each other. It could just look like a layers and layers of mealybug. They usually don't leave. So from the data that I just shared with you, we found that, yes, we can monitor for males using lure and trap cards. But do we see the same pattern throughout? That was one block data that I shared with you. We wanted to know if we see the same pattern throughout, different orchards in the Sanocan Valley. So to understand that, and also we wanted to know how many traps do we need per 10 acre to monitor for mealybug, because there is lure, there is trap, but there isn't a recommendation with it. So that's what for another study, what we did is we looked at 3 different trap densities, 2 lures in 10 acres, 1 lures in 10 acres, and 1 lure in 20 acre. And together we monitored 27 different locations. And the questions that we're wanting to answer is, do we see the same pattern of flight in all different locations in Kern County, Tulare County, Fresno County? And also, how many traps do we need per 10 acre? And this is what we found. We saw pretty much 5 to 6 peaks consistently in all the orchards that we monitored. Activity started April, May, second peak in June, third in July, fourth in August, September, October. Sometimes there was a peak in November too. That may have to do with weather. If weather is suitable, we might see a 6th peak. If not, we consistently saw 5 peaks in all the orchards, all the 27 orchards that we monitored. Did trap density matter? This data that I have shared with you over here, what it's showing is, these 3 different colors are 1 trap per 20 acre, blue line. Yellow line is 1 trap per 10 acres. And red line is 1 trap per 5 acres. That's, 2 traps per 10 acre. As you can see from these graphs, when you have 1 trap per 5 acres, the numbers seem, we were detecting, but not as many as when we had 1 trap per 10 acres. So our data the peaks overlapped for all trap densities, but data was more consistent when we had one trap per 10 acre. In Tulare County, in Kern County, in all areas. So based on these results, what we recommend is one trap per 10 acre would help you monitor mealybug throughout the season for males. Here is a trap card from 2 weeks ago. So when we were monitoring this field, we're looking for mealybug infestation. This is a field that we're monitoring for insecticide trial. We didn't see, good infestation. By that I mean we didn't see a lot of mealybugs on tree, on trunk and branches and on the inner canopy. But in the same field, we did find quite a good number of males caught in the traps, as you can see on this trap data of trap over here. All these brown spots are citrus mealybug males caught in the trap. I went to this field 1 week later, and this is what I saw. This is last week. So adult producing egg sacs. So give it about 2 weeks. By middle of June, just like we found in our data, we're going to see that second generation hatch, and that's going to move to the fruit. So to summarize biology, seasonal phenology. As for biology, immatures and adults feed, males fly. It takes about 4 to 5 weeks for a generation to complete out in the field. When does the activity start? Mealybugs are present all year round, but crowded activity starts in April. That's when we start noticing crowders from the overwintering eggs and laid by females that overwinter will start hatching and will start moving. How many generations? 5 complete generations and 6 male flights. So maybe 5.5 generation in the Sanokan Valley. How can we monitor for them? We can scout field, we can use male trap cards, and we can, look for signs of infestation, mealywax and egg sacs on trunk. And we can definitely use trap cards to monitor, especially early in the season. So moving on, for scouting and monitoring, when we're out in the field, we observed that citrus mealybug wasn't staying in the same place, especially when we're doing pesticide trials. We counted mealybugs on leaves, then, we go 2 weeks after they're not on leaves anymore. It's not because the insecticide worked, but because they moved. So we made a lot of observation and put together this slide for you on a calendar basis. So let's start with January. January to March in winter, when there is fruit on the tree, mealybugs are between clusters. On the navel end, under the calyx, they are on fruit. But if the block is already harvested and you have no fruit on the tree, then you might find mealybugs between, in cracks and crevices and under the thick canopy of leaves. That's where they live. Once the tree starts flushing in April, then mealybugs, the crowders from the overwintering generation, would hatch and move to the new flush. And this would be usually inside the canopy, especially on the suckers. They're not in outside canopy. You'd have to go in and look inside the canopy. And in July, the population, the second generation, then moves to the fruit and stays, in between clusters under the calyx. You can pop open a calyx to see if mealybugs are under there or not, and they stay on fruit throughout the season and get harvested with the fruit. So another way that you could determine if your field has mealybug or not is by looking for signs of mealybug infestation. That could be done by, walking the bins at harvest and looking for any signs of mealybugs, which is something like, this on the photo over here. Wherever the fruit are touching between cluster, if you see white stuff, even if you don't see any mealybug in there, but if you do see mealy wax present, that field probably has mealybug. And also, you could look on leaves and branches. You could look on the navel ends. Usually, if there is no mealybug, but there is a lot of sooty mold probably mealybug or other insects that produce honeydew. Okay, now the most important thing, right? The management. As with all other pests, for management, there are always multiple options. But because mealybugs are already a concerning pest, we want to focus on near-term management, and that's usually with chemical control. And that's what I'm going to focus, my next 20 minutes on. It is, however, very important to incorporate biocontrol and know about what biocontrol, what natural enemies exist and how all the management options that we're choosing is affecting predators and parasitoids that would attack mealybug. And that's something that I'm going to talk about too. Cultural control, pruning, hazing, part of all pest management. And I also want to touch base on ant management because ants and mealybugs would need to be managed together because we have seen evidence that in the fields where we have controlled ants, mealybug infestation is less. So let's start with the chemical control, developing citrus mealybug management strategies using pesticides. And this is a Citrograph article that was published in winter issue of 2025. You could just Google it and get it from there. I also shared links to arthropod management test articles. These are open access articles from all the pesticide trials that we do, published. Wherever you are, you can type arthropod pest management, go to the link, look for mealybug articles, and follow. Maybe Cheryl can put the links to these articles on the chat. And probably we'll send an email with all this information to you later on too. So let's talk about chemical control. When we started this work in 2022, we didn't know anything about what products work against citrus mealybug. We didn't have any efficacy data. The only product that UC IPM guidelines had was Movento. So our playing field was wide, and we wanted to know of all the products that are registered on citrus, which one works and which one doesn't. And we also wanted to test different group of insecticides. So we chose organic products, calcium polysulfide, azadirachtin, lime sulfur azadirachtin, and growth regulators, buprofezin, diflubengerone, pyriproxyfen, spirotetramat, selective products, acetamiprid, cyantraniliprole, flunicamide, epidopyropen. This particular trial was done targeting first generation. So we made our application on 29th of March when those crowlers from overwintering population had started hatching. Back in 2022, there was quite a bit a healthy population in the orchard where we're doing the trials, and we monitor population pressure for up to 4 weeks after the application. So on the graphs, what you're looking at is, first set is organic treatments, second is growth regulators, third is selective treatments. The first bar for each of the treatment is pre-treatment count, and that is number of citrus mealybug per sample, which is leaf. We counted citrus mealybug on leaf. And for all the treatments, what you can see, that second bar is 1 week after the application was made. From pre-treatment count to that first week, all treatments that we applied reduced mealybug population. Some provided longer-term control. For example, in organic treatments, calcium polysulfide, first week, great. Second week, the population started increasing. For azadirachtin, it continued to suppress population for up to 3 weeks. In fourth week, we saw increase. Same thing with growth regulators. All products suppress population for a week. For most of them, the mealybug started, coming back in second week, if not third week. The products that worked best for organic was azadirachtin. For selective treatments was acetamiprid and epidopyropen. For growth regulators, it was pretty much the same. There was no significant difference between treatments there. We repeated and also added new products during July application targeting second generation. And here also you can see treatments, organics, growth regulators, selective and broad spectrum. We looked at a lot of different products. Something that's different between July and March trial that I just shared with you is the sampling number of mealybugs. Because the mealybugs were now on fruit, they had moved to the fruit. We sampled fruit, and as you can see, your y-axis is pretty wide there from 0 to 300, and that's number of mealybugs per sample. There was huge variation because you'd see a fruit that looks like this that may have 300 mealybugs, and you'll see another fruit that has probably 2 mealybugs. So there was wide variation. But here also in July trial as well, all products that we tested reduced mealybug population for 1 week, 2 weeks, but in the following weeks, mealybug population started coming back. One of the things we also learned during this trial is counting number of mealybugs. How many is live on a fruit is not a good assessment. So we went with what percentage of fruit on a tree is infested with mealybugs. And when you look at that data, similar results as well. The product that suppressed mealybug population for up to 4 weeks was Sequoia, sulfhexaflor, danitol, Azadirect, dimethoate, lime sulfur. There wasn't a lot of significant difference because the error bars were overlapping, but some products did work better than others. For most of the products, it kept mealybug population pressure under 10% infested fruit for up to 2 to 3 weeks. Safina for up to 4 weeks, Movento for up to 3 weeks, lime sulfur for up to 4 weeks, Azadirect for up to 4 weeks, and danitol and Sequoia the same case. So learning from that, we learned that one application not enough. We need to make 2 applications to control mealybugs. So in 2023, that's what we did. We made 2 applications. First application in July, second application in August. And final evaluation was done in November, close to harvest, to see what percentage of fruit will still be infested with mealybugs. And what you're looking at is the combinations that worked were spirotratamate, fenpropathrine. But here the November final infestation in November was pretty high, about 30%. Spirotratamate, pyriproxyfen, that's Senstar, followed by Safina, up to 4 weeks, less than 20%. Organic opson, cinnamon, all in azadirachtin, and Center and Belief, also, a good combination in this trial. So now let's move on to natural enemies. The most reported natural enemies that are known to predate or parasitize mealybugs are listed here. The most common one is Cryptolaemus montageri. It's fairly common, especially in Southern California and Ventura region, but we haven't understood how well this species will establish and help control mealybug under Sanocan Valley condition. That's something that we'll be working on in the following years. The other predator that we see very commonly in citrus fields, and this is a photo that this photo in the middle is actually from a field, Cryptolaemus larvae, Chrysoporla larvae feeding on citrus mealybug. And then there is quite a bit of parasitoids, Anachidas, leptomastics, and coccidogenoids are also commonly occurring predators, parasitoids out in the field. We have collected infested mealybugs, parasitized mealybugs from the fields and reared them out. All these photos that you see line at the bottom are actually field photos of parasitized colonies. Here are some of the results from an ongoing study that Kelby Keeling is doing to understand what kind of background mortality is going on, what kind of natural enemies are in the Sanocan Valley. And as you can see from these graphs, and these are from different counties, in Ivanhoe, in Tulare County, in Fresno County, also in Fresno County, The blue bar is anagyris, the blue line is anagyris, and all these graphs. And as you can see, anagyris activity increases in July, and pretty good anagyris parasitism going on in all different orchards. The table that I'm sharing here is something that they observe actively parasitizing mealybug colonies out in the field, and out of those, nearly 7%, 7 out of however many times, 7% of the observation rate, they always saw green lacewing larvae feeding on mealybugs. Anagyris also another common parasitoid. So for us, as someone who is developing management strategy for citrus mealybug, another thing to keep in mind is insecticide selection and its effect against natural enemies. Here is a lab study that we did looking at different effects of different products on Cryptolemus adults and larvae, and also on anagyris adults. And as you can see, Cryptolemus adults pretty tolerant. Most of the insecticides didn't affect its survivorship. Acetamiprid was the one that affected survivorship 48 hours after exposure. Same product, more harsh on larvae. Also on anagyris, of all the products tested, acetamiprid was toxic. After 48 hours, you have less than 10% survival. Abamectin also toxic. Epidopyropen also toxic. The relatively safer products were azadirachtin and spirotratamide. Now, this study is pretty conservative because we are exposing these insects without any food to a leaf that's treated with insecticide for 48 hours. That doesn't usually happen out in the field. We are following up in this year and in the next seasons with a residual study to see which products, how long after you have sprayed a field with insecticides, it's okay to release natural enemies. But from this, you can get an idea that, hey, acetamiprid is relatively more toxic to anagyris adults compared to azadirachtin or spirotratamide. Okay, now let's look at some videos that I took last year out in the field, and hopefully it plays. Oh yeah, it does. So there is an anagyris adult laying an egg on CMB third instar. And then they're here in the video, it's the same fruit, actually, the same field actually, not the same fruit. But look at this field ant coming over here and then chasing this parasitoid away. That's an anagyris that's running away. So another thing that I'm sharing with you here is a study that Kelby Keeling is doing as a graduate student, looking at what kind of ant species are we seeing associated with mealybug? What is tending mealybug? And this is from her research. The pie charts down below are the ants that were collected on the floor, on the ground, and pie charts on the top are the ants that were collected actually tending the mealybugs. And there were quite a variety of ants tending mealybugs in different areas. In Sanger, where Argentine ants had established, they only found Argentine ants tending mealybugs. But in other areas, where Argentine ants were not present, there was quite a bit of variety of ants. Now, the next question is, does ant control help mealybug? And our data says so. This is from 2023 and 24 study where we looked at citrus mealybug infestation in fruit as represented by the bar charts in fields that were ongoing ant treatment. And as you can see from the dotted lines, wherever there were less ants found, there was no ant control because there was still quite a good amount of ant activity. But wherever there were less ants present, our mealybug infestation was 50% less. And when we tallied those numbers, there is 70% correlation between number of ants found and percentage fruit infested with citrus mealybug. And also on this, another bar over here, the pink line is the ant-free trees, and the blue lines are the trees where ants are tending mealybugs, and we saw more parasitism in trees where there were no ants compared to the trees where there were ants. Again, going back and reconfirming that ants are chasing mealybugs, and ants are tending mealybugs, protecting mealybugs, and chasing natural enemies away. Okay, now moving on to cultural control. There hasn't been a lot of studies done with cultural control and mealybug, but maintain good sanitation, remove old infested fruit and branches when possible. Scout the field looking for early signs of infestation. It's always easier to manage when the population pressure is low. Prune the trees. They like that thick canopy environment, so open up the canopy because mealybugs like dense foliage, and that also allows for natural enemy activity if you keep your canopy fairly open. Okay, on to my last slide. So mealybug best management practices, activities, and this is, for California citrus growers. Activity starts in March when crowders from overwintering females start moving to new flush. That's when you can start monitoring. If you have a known infested like field, usually more than 10% of the fruit at harvest had some sign of mealybug activity, then you might want to watch that field and treat early in the season, because treating when the population pressure is low is going to help you manage population throughout the season. Scouting is season dependent. In January and March, look for overwintering eggs and adults between fruit cluster. From March to May, June, they are on young flush. From July to November, December, they're on fruit. And you can also monitor for males using pheromone lure and trap. For management, always target early generations. First, second generations are way easier to manage because there is less numbers. It's all a numbers game when it comes to insect management. And also there isn't overlapping population. So populations are more synchronized, easier to manage. From pesticide trials, 2 applications may be necessary. You could either tank mix 2 products, insect growth regulator with an insecticide that provides a good control, or you could do 2 application, within a period of 2 to 3 weeks. Organic option, as a direct in cinnamon oil, works fairly good. Lime sulfur works really well. And another thing you want to keep in mind is rotate products to avoid resistance development and get a good coverage, because mealybugs are inside the interior canopy. Choose products that are safer to natural enemies. And if you're releasing natural enemies, maybe wait a week or 2 after insecticide application to make releases. In future, we'll continue to focus our study on, fine-tuning insecticide application, timing, understanding residual toxicity to natural enemies, and ant management, and most likely mating disruption. That's all I have. This research is totally supported by Citrus Research Board, but I want to acknowledge Citrus Research Board, all the pesticide trials. I want to thank chemical companies for supporting the trials, donating the products, everybody who is listed over here for all their help, collecting most of the data that I presented to you today. Okay, that's all I have to say.
Thank you so much. We have a couple of minutes, so I had a couple questions in chat I wanted to ask you before we go ahead and put the link in the final in the chat to the test. But one of the questions in the chat was, do oil sprays have any impact on citrus mealybug?
Yes, they do. I didn't mention it, but all the insecticides were applied with 1% oil and surfactant, either exit or new film, depending on what products we're using. And they do oils, just like with any other insects, you need to get a good coverage. Oil alone probably won't give you the level of control you would need, but it does have an effect. And especially if you're mixing it with insecticide, it helps get a better coverage.
Okay, thank you. And then just probably one more quick question before we do the test link. Why would lower trap density provide more accurate data?
Um, well, we only tested 3 trap densities, and we got consistent results with 10, 1 trap per 10 acre. And when you're putting like 2 traps per 10 acre, we're not catching more. So that goes to say that lure is probably fairly effective and mealybugs can travel quite a distance. Um, Based on what we have done, we only tested 3 trap densities, so there isn't a lot of information to say that lower trap densities would provide a better monitoring compared to higher trap densities. But 1 per 10 acre seems to do the same job as 2 per 10 acre.
Okay, thank you. And so, Sandeepa, thank you for presenting today, and I appreciate you answering some of these questions in writing. We appreciate you taking the time to present for us today.
Thank you, Cheryl. Thanks for the opportunity to talk to everybody. Happy to help.